Hi guys and gals, let's continue our discussion of biotechnology. In our previous lecture recording for this chapter, we talked about DNA, RNA, and how to use DNA and RNA to make proteins. In this second recording, we're going to focus on the biotech itself. How we can clone DNA, how we can copy DNA, how we can analyze DNA, and then how we can modify organisms using their genetic code. So first, let's talk about genetic engineering. When we talk about genetic engineering, we mean altering the DNA in an organism through recombinant DNA technology. And when I say recombinant DNA technology, I mean that we're going to be combining pieces of DNA from multiple organisms. When we do that, when we use recombinant DNA technology for genetic engineering purposes, we make a transgenic organism. A transgenic organism is going to cross the species line. It's trans, and it contains genetic information from different species or organisms, all combined into one. To do this, generally speaking, we're going to use biotechnology. Biotechnology is based on already present systems in nature. To do these processes, we have found techniques that are being done by bacteria and viruses and other organisms, and we mimic them and copy them in a lab setting to specifically guide and direct changes to the organism's genome. So first, let's talk about DNA sequencing. Before we can do any major kind of biotech, we need to first know the sequence of the nucleotides in the DNA. Back in the dark ages, the 1970s, this was done manually using dye terminator substances. In other words, we would try to duplicate the genome, and we would have special nucleotides that would end the duplication process, and these special nucleotides would glow different colors. And then we would run the genetic fragments through a gel to separate them based on length, and then look at the different colors they were glowing, and then know the order of the genome. It took forever. Now it's much faster. Now instead of using dye terminator substances, we still use those dyes, but we no longer have the terminator substance. Instead, we are going to reproduce the genome, and then it will glow different colors. And those different colors are read by a laser, and instead of having many hundreds of thousands of small fragments that need to be arranged in order from longest to shortest, we instead have one fragment that's read linearly by a machine that looks at the color each nucleotide glows. So here's a picture of a DNA sequencer, and that DNA sequencer looks at the color wavelength of light that each region of the genome will glow. And based on those colors that they glow, the sequencer is then able to say what nucleotides are present in the DNA fragment. To amplify a segment of DNA, we go through a process known as Polymerase Chain Reaction. And polymerase is a generic name for an enzyme that makes polymers. And this chain reaction is used to clone very small segments of DNA. The polymerase chain reaction used to take place using water baths, and you would have to take your samples and put them in hot, cold, and medium temperature water baths. Now they're done in a simple machine known as a thermocycler, which shaves hours and hours off of the sequencing time. I should say days and days. Before we can do any other bit of DNA work, we need to be able to amplify the specific chunk of DNA we're working with till we have a concentration that's high enough to be detected by our machines. To amplify our specific sections, we use PCR. We first start off with the segment that we want to amplify. We heat it up to its melting temperature, and by heating it up, we cause it to become single fragments. And those single fragments are then going to have a primer strand of DNA attached to it. That primer strand of DNA determines what will be amplified. And then once the primer is attached, DNA polymerase is then able to synthesize a new daughter strand. Then we've doubled the number of DNA molecules. And we repeat this process over and over and over again where we heat them up to separate the double strands to make single strands, attach primers, DNA polymerase makes a new strand, and we keep duplicating over and over and over again. Generally speaking, we go through about 30 to 40 rounds of replication until we get a concentration that is high enough for us to work with. After we've gone through many rounds of replication, you can take those DNA fragments and separate them using an electric current and a gel. This is known as gel electrophoresis. And you can use this process for DNA fingerprinting. You can take DNA from different individuals and compare the fragments of those DNA to see what they have similar and what they have different between them. During this process of gel electrophoresis, you'll take your gel that you run your electric current in, and you'll load up your DNA into sample wells within that gel. And then you'll run an electric current through the gel that pushes the DNA away from the sample wells. We typically are going to need a ladder lane. This ladder lane includes DNA fragments of known sizes so that we have a scale or a frame of reference for these other DNA fragments. And with those other DNA fragments, you can take a sample from your crime scene, and this sample had these specific 2 bands to it, and compare it to suspects. This suspect A doesn't match the crime scene sample. However, suspect B does match the crime scene sample. Therefore, we can assume that suspect B was at the crime scene. We can also use DNA fingerprinting to determine paternity, to determine the identity of unknown remains, and to also look for relationships between species or within a population. During gene cloning, we are going to recombine bits of DNA from multiple species or multiple sources. This recombinant DNA technology allows for us to clone genes in other organisms. Generally speaking, we're going to use lots of bacteria in this process. We took the gene for human insulin and inserted it into bacteria. To do this, we needed to slice the bacterial genome and slice out the gene for human insulin. To slice DNA, you need to use a special enzyme known as a restriction enzyme. Oftentimes, they're referred to as a restriction endonuclease. And this restriction enzyme is then used to cut out the gene that you're interested in and put that gene into a carrier molecule that carries it into the other organism. The most common carrier molecule used in this process is called a plasmid, which is a little circle of DNA. This carrier molecule, generically speaking, is called a vector. Occasionally, a vector other than a plasmid is used. Notably, you could take a piece of gold, fill it with microscopic holes, and then use a helium gun to blast that piece of gold, kind of like a shotgun shooting birdshot, to blast that microscopic piece of gold into the cell to insert the DNA or your favorite gene. That process, though, is very expensive and not as effective as designing a circular piece of DNA, a plasmid, to insert your favorite gene into the organism. After you insert your favorite gene, you need to use DNA ligase to snip the ends together to seal it in, and then get your vector into the cell. So you can take from a human being the gene for insulin, and then take that gene and insert it in a small plasmid or circular piece of DNA. So you have a modified piece of DNA or plasmid that's then put into a bacterial cell. That bacterial cell then reproduces over and over and over again. And now you have lots of little bacterial cells that are making the protein for the gene you inserted. And those bacterial cells are then secreting human insulin, and we then have a new product that we can use. Important uses of transgenic organisms for human being include making insulin, human growth hormone, other proteins that have very medically significant implications for human beings can be made with transgenic organisms. We've also designed bacteria to help clean up from oil spills. We've also designed other organisms to be less likely to die in a drought, or to be pest resistant, or to resist herbicides as well. So we can use transgenic organisms to produce human medicines. We can use them to design fish that grow bigger sooner. We can use them to design food products that have increased vitamin content. And we can use transgenic organisms to design bacteria that clean up oil spills. If we look at transgenic plants in particular, and I spent a lot of time working with transgenic plants in the research lab that I worked in, we can design these plants to make human proteins so that we could potentially have a plant act as a human vaccine or human medication. We can design plants that resist herbicides or insects or resist frost so that we can have increased crop yields and higher rates of food production and ideally less hunger in the world. If we look at transgenic crops in the United States, corn, soybean, cotton are commonly, almost always going to be transgenic now. As recently as 2011, and this number has only continued to go up, the vast majority of corn and soybean in the United States is going to be genetically modified. These genetic modifications mean that you can spray an herbicide on the field, and that herbicide kills all the weeds, but doesn't kill your crop. I grew up in rural America, and I remember before these transgenic plants were available, a common job was weeding the field by hand. And I would have to walk up and down every row in this cornfield and pull out all the weeds. And I'd walk up and down every row in the soybean field and pull all the weeds out of the field by hand. It took forever, and it was a highly inefficient process. By being able to have herbicide-resistant plants or crops, we now no longer have to spend hours and hours of our lives weeding the field by hand and have higher crop yields as a result. The same can be true for designing plants that resist insect or herbivory. We can design plants that produce natural insect toxins. Here's a plant, a control plant that did not produce the insect toxin. Here's a plant that was designed to naturally produce the insect toxin. And understandably, the plant that was toxic to bugs grew much larger than the plant that was not toxic to bugs. We can have many different uses for our transgenic organisms. We can design crop species that can grow in high salt environments. We can design crop species to have increased carbohydrate or increased protein or increased oil content. Whenever we make a transgenic animal, unlike a transgenic plant, to make a transgenic animal, we need to insert the gene into the egg that's being fertilized. So we need to insert the modified gene into the gamete in order to guarantee that that organism as a whole will have a modified genome. This can be done relatively simply by inserting that vector into the gamete. When we go through this process, there are many different uses for it, one of which is to make pharmaceuticals in the milk of farm animals. There was an excellent article I read recently that involved modifying goat milk to produce spider silk proteins and then harvesting the spider silk proteins from the goat milk to come up with an alternative for Kevlar. With large organisms, you can design those organisms to grow faster. You can design them to produce more meat or more of that product that you're hoping for. When we work with mice, generally speaking, we design mice to have specific genetic diseases so that we can then study that disease in detail. A common method is, right now, is to modify the pig genome so that that pig genome is more similar to the human genome, and we can transplant organs from a gene into a human being. That is known as xenotransplantation, taking organs from one organ species and putting it into a different species. Here's a diagram showing how to make a transgenic organism. You need to first modify the genome and then insert that genome into the gamete or the egg cell of the organism, and then that offspring grows with the modified gene in every cell of its body, and that modified gene then causes it to make that organism to produce whatever protein that gene was coding for. And then you can breed that organism and then produce a bunch of goats, for example, that produce milk that contains the specific protein you are looking for. In this case, human growth hormone. So, guys, girls, concept check. Which of the following techniques specifically amplifies a sequence of DNA? Is it DNA fingerprinting, transgenic plants, polymerase chain reaction, or crude oil consuming bacteria? Go ahead and pause this video and get me an answer. 5, 4, 3, 2, 1. The correct answer is C. PCR specifically will amplify a section of the genome. Moving on. What can we do now with biotechnology? Fairly recently, within the last 20 years, we concluded our Human Genome Project. This Human Genome Project involved sequencing the complete human genome. A human being has approximately 23,000 genes, or 3.4 billion base pairs. The human genome was finished being sequenced in 2003, and since that process was done, we've now started data mining the Human Genome Project. We're now trying to find how genes can change from individual to individual. There are many small changes within the human genome from individual to individual. These are known as polymorphisms within the DNA. Our genome, though, size is not going to be correlated with the number of genes or the complexity of the organism. Some less complicated organisms have enormous genomes. Other more complicated organisms will have smaller genomes. Within the human genome, there are large regions of the genome that have an unknown function, that do something that we don't know about yet. So we can look at functional genomics. We can look at what those 23,000 genes do in our bodies. These 23,000 genes within the human genome make up less than 2% of the overall number of base pairs within our human genome. Most of our human genome right now doesn't have a specific cause or purpose tied to it. Now, since this genome has been sequenced, we're starting to figure out what these non-coding regions of the genome do. And these non-coding regions of the genome have been linked to lots of regulatory functions that help to regulate the genes of the genome. A term you may read about is when reading about regions of the genome that are not coding regions is junk DNA. Junk DNA is falling out of favor right now because we're starting to realize that these large regions of the genome that we thought didn't do anything are now going to regulate the transcription of the other genes within the genome. We can also perform comparative genomics. When we perform comparative genomics, we are going to look at the genome of one species and compare it to another species. And by comparing the gene sequences between species, we can help determine an evolutionary relationship between species. So when we look at our functional comparative genomics, we can look at how the genes between organisms are the same, how they're different, and how that genotype will influence the phenotype of the organism. Some new endeavors in the horizon are proteomics. Proteomics looks at analyzing the structure of the proteins of the cell. Proteomics is difficult because between different cell types, we can have different amounts of messenger RNA editing. Protein concentrations can be different from minute to minute, from cell to cell, and the amount of regulation that occurs for that protein differs from cell to cell. As we learn more about the proteins of the cell, we are starting to better design drugs to help us not experience disease states. I should also throw in here microomics. Microomics is the study of the microbiome, or how the bacteria in our body, the microbes in our body, interact with the human cells of our bodies to influence our overall health. And then we also have bioinformatics. Bioinformatics is the field that looks at using computer analysis to analyze the large number of data points we get by sequencing an organism's genome. To modify an individual's genome, we need to apply gene therapy techniques. The new kid of the block that didn't make it into this textbook is known as CRISPR editing. And you can go ahead and Google that to read it up on it on your own. CRISPR editing will not appear on your test. It's the newest kid on the block. With our gene therapy, we can have insertion of genetic material into a human cell to treat a specific genetic disease. We can have ex vivo therapy. During ex vivo therapy, we take a sample of the patient's cells out of their body, modify them, and then put them back in their bodies. With in vivo therapy, we are going to modify the human's genomes while those cells are in the patient's body. Gene therapy is starting to become quite successful as a new novel technique for treating cancers. So, if we look at our gene therapy, an ex vivo method is to remove the cells from the patient, modify the cell, take that cell and put it back into the patient to help relieve the disease state from that patient. Oh, my bad. Patient's body modified and then put back into the patient's body. Is it bioinformatics, microbiomics, proteomics, or ex vivo therapy? Go ahead and pause the video and get me an answer. 5, 4, 3, 2, 1. The correct answer is D, ex vivo therapy. If you have any questions for me about this lecture material, please feel free to shoot me an email, post it on the discussion board, or swing by my office when you are on campus. Happy studies!